Our next speaker is Prof Robert Langer. Prof Langer is joining us virtually and will share about the topic from nanotechnology to mRNA therapies and what's to come. How overcoming skepticism and barriers led to new vaccines and ways to tackle global health challenges. The Q&A session will be moderated by Prof Ali Mizrahi. Prof Langer, please.
So why don't we? Let me go over how I got started. So I was a chemical engineer, and all my colleagues would go to chemical engineering jobs. Like, thank you for the slides. They, they 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 would go to oil companies. So I thought I would do that too. So I wrote. So a lot of the oil companies would come to MIT, and I actually got 20 job offers from oil companies. A lot, even about 4 or 5 from Exxon alone. And I wasn't that good, but they just had so many openings. So one of the, but I felt when I went to these companies that the impact I'd make wasn't that great. They'd say you could spend your life improving the yield of some of these chemicals by 0.01%, and that would be what you do. And I thought I didn't want to spend my life doing that. And so I started thinking of other jobs I might do that could help people. And I was very interested in education. I helped start a school for poor high school kids. And I got very involved in creating new chemistry curricula. So I wrote, one day I saw an ad at one of the universities to do that. And I wrote them applying for a job, but they didn't write me back. But I tried about 40 different colleges to do that, and none of them wrote me back. So that wasn't going so well. So I thought other ways I might help people were by using my chemistry and engineering background in medicine. So I wrote to a lot of hospitals and medical schools, and they didn't write me back either. But then one day I walked into the lab, and one of the older scientists said to me that, Bob, there's a surgeon in Boston named Judah Folkman, and sometimes he hires unusual people. Um, he thought very highly of Dr. Folkman. I won't say what he thought about me, but I wrote Dr. Folkman a letter, and he offered me this job. And he had a very unusual idea. This was 1974. And if we could go to the next slide. Could So this was his idea. His idea, this is a picture of how a tumor grows. If you go on the lower left, the idea is the tumor is somehow abnormal. It grows as a three-dimensional mass, and and if you go to the center, you can see that the center bottom, it really doesn't grow beyond about 1 million cells. So, but the way the tumor solves this issue about getting bigger, see, it's a nutrition problem. Cells in the center can't get nutrients. So what he suggested was tumors make substances called TAF, or tumor angiogenesis factor, and that causes the surrounding blood vessels to grow to the tumor. And then when you go to the upper right, notice how now the tumor gets bigger and bigger. And on the utmost right, it's very vascularized, and the tumor can spread through those blood vessels. That's a process called metastasis, and that often kills people. Um, so what? So this was his idea that this is how tumors grew, and most people didn't believe him. I didn't know anything about biology, so I thought it was a great idea. Uh, but there was no way to study this. So we had to develop what's called a bioassay. And there was no way to study blood vessels then. All these molecules that we were isolating to try to see if they could stop blood vessels, they were big molecules, and we had to deliver them for a period of time. And no one had done that before. And Dr. Folkman, he was on a board of the only company in the world, ALZA, that was working on this. And so he went and asked them, could they help? But if we go to the next slide, what they said is that you could never do that. Oh, this is just, this was the assay we wanted to use, that you'd put a tumor in the eye, and then you'd have a tiny nanoparticle or microparticle delivery system that would deliver these large molecules that I was mentioning. And like I say, he asked these people that were real experts in this, could they ever help us? But they all said, next slide, that they couldn't, because the molecules were too big. If we could go to the next slide. Yeah, they just said they were too big to get out of these, through these solid materials. So anyhow, if we were going to solve this problem of blood vessels, I had to solve the problem of delivery. So I spent 2 years in the laboratory experimenting with different materials and different designs. I found over 200 different ways to get this to not work. But eventually, if we could go to the next slide, I was able to make tiny little micro or nanoparticles. You see a picture of one on the left, cut in half on the right. And what we showed on the next slide is that you could use this approach to deliver molecules of any size. This was a paper in Nature we wrote in 1976. So you could release molecules of any size, even nucleic acids like mRNA or DNA, for the very first time. But when I first presented this work, it was really ridiculed. I was 27 years old, probably like the age of some of you, and I was the youngest person at the scientific meeting of chemists. And I gave this talk, and when I got done, I thought I didn't do that badly because I didn't forget too much what I wanted to say. So I thought all these older people, being, nice people, they would want to encourage me, this young guy. But when I got done, I stepped off the podium, and a whole bunch of them came up to me and they said, "We don't believe anything you just said." If we could go to the next slide. Again, they said that the large molecules were too big to diffuse through little cages of like nano or microparticles. And also they were pointing out that and this is true, I used organic solvents like ethanol and methylene chloride to do these encapsulations. And they said that would destroy all these molecules I put in. So that wasn't so good. And actually things kind of went downhill for me from there, if that were possible. I wrote grants to try to fund some of our work, and my first 9 were rejected very, very soundly. I also, I really liked being a postdoc, but a lot of my friends said, I should apply for faculty positions. And like I said, I was a chemical engineer. So I did. I applied for faculty positions. No chemical engineering department in the world would hire me. They all said, this bio stuff you're doing doesn't make any sense for a chemical engineer. You should be doing oil or energy. So that wasn't so good either. Finally, I got a job in the nutrition department. But the problem there was the department head, he was what I'll call a benevolent dictator kind of department head. He liked me, so he offered me a job, but he didn't bother to ask anybody else in the department what they thought. Now, that might have been okay, except for one thing, which was the year after I joined the department, he left. So a lot of the senior faculty decided to give me advice, and their advice is I should leave too. If we could go to the next slide. My friend Mike Marletta, he's a very famous chemist who was head of Berkeley's chemistry department. He was reflecting a few years ago when I got some award, what it was like to be with me in 1979 at MIT. And here's what he said. One evening, I went to a faculty dinner at a Chinese restaurant with myself and some senior MIT professors. He said, a senior scientist sat quizzing us while smoking a cigar. He said, when the older scientist heard my concepts for drug delivery, he blew a cloud of smoke in my face and said, you better start looking for another job. So I had a very inauspicious beginning, but I really believed in what I was doing. I could see a lot of this with my own eyes, that we put different dyes in and they of large molecules and they would come out. And so we kept at it. And I mentioned to you that one of the problems we wanted to solve was the blood vessel problem. So I also, at the same time, was isolating different substances, and I had different fractions, and so I would put them in these little nano or microparticles, and we had that rabbit eye assay. And almost every one we isolated did not work. We did about 2,000 eyes, but one did. And let me just show you, and I apologize for showing such a bloody slide, but let me just show you on the next slide what happens. So if we put, these were the cornea, if after a period of time you put a sham or inactive fraction, you get what you see on the left, say after a couple of weeks. Ultimately, those tumors would kill. but we sacrificed the animals before. But when you put this one inhibitor in, look on the right, and we repeated this 20 times, the vessels are lower, they're sparser, and they avoid the tumor. We published this in Science in 1976, and that really, I like to think, began to change people's thinking. Nonetheless, it still took 28 years from this Science paper before the first blood vessel inhibitor was approved by FDA. And that took great work from Genentech and other companies. But the next slide was kind of striking about what ultimately happened. And this may be a little hard to see, but what you see is that starting in 2004, 28 years after our paper, one after the next blood vessel inhibitor is approved by regulatory authorities. Avastin, the first of these, is one of the 2 or 3 largest selling drugs, biotech drugs in history. And not only can you treat all kinds of cancer this way, as you see in the right, but eye diseases like macular degeneration, one of the leading causes of blindness in the world due to abnormal vascularization, there was no pharmacologic treatment before. Now drugs like Eylea and Lucentis are blockbusters, treating many millions of patients with these diseases. So, like I say, this would lead to all these inhibitors. But I also thought that the drug delivery work itself would be useful. So, one of the things Dr. Folkman said to me one day is, Bob, we should file a patent. And Children's Hospital had never had a patent before. I didn't either. So we filed the patent. 5 years in a row, the patent examiner rejected it. And the people at the hospital came to see me and they said, Bob, you should just give up. This guy's never going to allow it. But you can tell by now, I don't like to give up. So I started to think, how could we convince the patent examiner to allow this, legally, of course? And science really wasn't working. But as I mentioned to you in the beginning, that when we first started doing this, everybody told me this was impossible. It could never work. I wondered, maybe someone wrote that down. So now in 1982, I did a science citation search to see who cited our 1976 Nature paper. And I found it's always interesting to see what people say about you. At any rate, let me just show you one article. Next slide. So I had no idea this was written, but it was 5 of the top chemists and engineers in the world in 1979. I'll just read you what they wrote, describing this really embryonic field. They said, generally, the agent to be released is a relatively small molecule with a molecular weight no larger than a few hundred. One would not expect that macromolecules, for example, proteins, could be released by such a technique because of their extremely small permeation rates. However, Folkman and myself have reported some surprising, I thought surprising, that's a really good word for a patent examiner, results that clearly demonstrate the opposite. So I showed this to the people at the hospital. They showed it to the patent examiner. And he said, gee, I tell you what, I will allow this patent if Dr. Langer can get written affidavits from those 5 people that they really wrote it. So I wrote them, and they were all nice enough to write me back. They really wrote it. And the examiner did allow this patent that you can see on this slide. Now, with that patent, we actually licensed it, and I consulted for a couple of very large multi-billion-dollar companies. I was so excited. They gave me grants, and they were, giant companies. And I thought, gee, something I did might help people someday. But, they worked on it maybe for a year, and they just gave up. And then one of my colleagues, Alex Klevenoff, said to me one day, Bob, said, we should start our own company. So we started this little company, Enzotech, which later merged to become Alchemies. And we also started other companies and also licensed things to different companies. And that has led to many, many products, some of which are shown on the next slide. And there are all kinds of things that you can inject into the body that can treat cancer, heart disease, schizophrenia, type 2 diabetes, even like what are called drug-eluting stents, which is in the bottom right, which is one of the major treatments for heart disease. So hundreds of millions of people now use these every year. But going back to the academic lab, I could still see that there were lots of issues to solve. When we made most of these that you're just seeing here are like microspheres or implants. As we make things smaller and smaller, you could get release like we showed in that Nature paper. But sometimes what would happen is the little particles, like the nanoparticles, they'd stick together and aggregate. And also they could get eaten by macrophages. So we had this idea, next slide, of making a special coating. We did this with Vladimir Torchelin and others, and called polyethylene glycol. This will end up being one of the 4 key components in all lipid nanoparticles. But now, this is almost 30 years ago. So we published this in Science. And let me just show you a little bit of data. The next slide shows some atomic force micrographs. The next slide just shows you that they really are nanoparticles. Next slide. The next slide is quasi-elastic light scattering. And what's really exciting is shown on the next slide, that when you put an orange dye in them and you have a macrophage, if you don't have PEG, look on the upper left. They're all eaten. They're all in the cell within an hour. But when we coat them with PEG, they're hardly taken up at all. And there are different designs we use. So that was very exciting. And then we thought, well, we could even do targeting. And the next slide is the sort of that idea that, and Omid Farrakhanazad did a lot of that. And by the way, sometimes I feel I don't explain this that well. So I want to show you a video on the next slide that Nova, the TV show, did. If we could turn on that next slide in video.
He starts with a nanoparticle of anti-cancer drug. That gets encased in a plastic that releases the drug over time. That in turn gets a special wrapping that disguises the package as a water molecule to fool the body's immune system. And last but not least, the address where it should be delivered, a key that will only fit the lock of cancer cells.
Now, I should tell you that all the clinicians I work with tell me it doesn't blow the cell up quite like that. But at any rate, so that's what we thought about. And then we also, with Dan Anderson, who was one of my postdocs, decided to do work on the 4th component. Like there's 4 components in lipid nanoparticles: cholesterol, structural lipid, now a PEG lipid, because in part of what I just showed you, and then like what's called an ionizable lipid. But when we started, there were very few lipids that people used. So we came up really with the first ways of doing what I'll call combinatorial synthesis of lipids, where we could make thousands of them. And this was the wish list shown here, that you could do it in a single step. And part of what we did is use certain types of chemistry, illustrated on the next slide, like a Michael addition. And by doing that, you could react diamines, and basically you could make lipid-like materials. And you could get enormous chemical diversity by changing the R group. And the next slide or two will show you that. The next slide just shows large variation in the R group. And you can also change the tails and number of tails, as shown in the next slide. And so you get enormous diversity. So we literally made thousands of tiny little lipid nanoparticles. And on the next slide, this is all work we did in collaboration with El Nilam, with Akina Kink, who was one of my students, but then went there to lead their program on rare diseases for ATTR amyloidosis. So you can get a dose response curve. You can get this is factor 7 in vivo, but the next slide shows you get a dose response curve. As you increase the amounts, you get more and more knockdown. The next slide just shows you can get a real treatment possibly for atherosclerosis by knocking down ApoB with some of these. And you can see that here. And you can give these, we find you can give these sRNAs repeatedly. You don't have to just you can give it once and it might last for a month. But the next slide just shows you can keep doing it over and over, which was exciting. And then, of course, Dan and I, you want to do better. So what we did is we could take the initial libraries we had, but then we could improve them. And an example of that shown in the next slide, that we could take the amines from the original library, take the best ones, but now use an epoxide chemistry to make 150 new compounds that we could test in vitro and then in vivo. And let me just show you the data as we improved the efficacy by this 4th lipid. So what you're seeing on the left is the LNP01. That's what I showed you earlier when you get knocked down. But you can go maybe almost 2 orders of magnitude better by doing the epoxide chemistry one I just showed you, and that's in the middle. So you get this dose response curve, but you get some knockdown with as low as 0.003 mg per kg. And that translates to monkeys, next slide, non-human primates. And again, you can get this with not only, with non-human primates, very significant knockdown. And over a 10-year period, our cells working back and forth with Alnylam and some of their collaborators in Canada, we'd go back and forth, improving the potency. And the next slide just shows you what it shows you. It started out with, in 2004, you could see, if you go to the right-hand side of the screen, you get these first-generation nanoparticles, but you can keep improving them and improving them. And by 10 years later, you're up to about 4 orders of magnitude better, more potent. So that was very exciting. And I mentioned to Kinnikinck, and he led this effort. He was my student at Alnylam. And what that would ultimately lead to is shown on the next slide, which is Onpattro. This was the first lipid nanoparticle with a nucleic acid, or first nanoparticle with a nucleic acid in it. It's used to treat a rare disease, ATTR amyloidosis. It's a nerve disease, and it's been really a wonderful thing for those patients. So at any rate, that was 2018. Now, of course, we started a lot of the work a lot before that, but even back in the 70s. But in 2010, now go back in time, myself, Derek Rossi, Nubar Fayan, and Ken Chen, we had this idea that, well, okay, siRNA, that knocks genes down, but messenger RNA, you could get the opposite effect. You could get gain of function. The next slide is really just a central dogma of molecular biology, that DNA makes RNA makes proteins. Well, by this time, 2010, there were enormous number of protein therapeutics that had come about in the last 30 years. But I'm sure you all know it takes a long time to manufacture them. You need these giant cell factories. In the case of some vaccines, you need eggs. It may take a year to do it. Here, we thought if you could make messenger RNA a drug, what you could do is you need to protect it. That's where you need the nanoparticle. But if you inject it in the body, body's the factory. The body will make the protein. So you could do this very, very quickly. In fact, we started this company in 2010. By the end of 2019, we had 13 different products in human clinical trials. And then COVID came. Next slide. And before I do that, I just want to mention there's a lot of other diseases you could treat too, like intracellular or membrane-bound proteins you could make this way. At any rate, like I say, we worked on this at Moderna for about 9 years, and then COVID came in late 2019, early 2020. And let me just show you how fast Moderna was able to make the vaccine. It was pretty much unprecedented. Next slide. So the Chinese scientists published the virus genetic sequence on January 11th. It took less than 2 days to finalize the design, put it in the nanoparticles, and within a month, we had shipped batches to our clinical partner, NIH, for testing. The first patient was treated in what's called a phase 1 trial, literally about 63 days after we started, 64 days. And by May 19th, we had the data on the phase 1 trial. Just so you know, in case people aren't aware, when you do a clinical trial, usually you do escalating dose to find what the highest safe dose is. So what we did is we had that data by May 19th, and we could see what dose was safe, but also you can get significant antibody titers, and you could find out if the vaccine was likely to work. So, when Moderna announced that data, I thought that was pretty exciting. Nonetheless, the news media and many scientists were just like they were always critical of me when I started, they were very critical of Moderna. The next slide shows the Boston Globe, my local newspaper, their headlines on May 19th. And their headline was, "This is not how you do science." And you can see whose picture's on the cover, front page, me, which was unfortunate, but that's the news media. At any rate, so I want to go just 6 months after that. So after you do a phase 1 trial, then you do a phase 2 trial, and finally you do a phase 3 trial. So we started that, and by November, That trial, which had 30,000 patients, was done double blind. We broke the code. And I remember being in the boardroom. So 15,000 get placebo, 15,000 get our vaccine. And it was remarkable when those data were announced. Not a single person of the 15,000 died. Not a single person of the 15,000 even went to a hospital. And it was 94 to 95% effective. And of course, next slide, it's now been used all over the world. Another one in nanoparticles by BioNTech and Pfizer also has been used all over the world, probably several billion people. Let me just show you quickly some of the data. Next slide. So this is, showing you what happens between unvaccinated on the top, vaccinated with no booster, and then vaccinated with a booster. Let me show you on the next slide the effectiveness of the different vaccines. The Moderna one, 94%, Pfizer and BioNTech, 91%, Johnson & Johnson, 66%. And then that got pulled from the market. There's multiple articles published in the past year about the efficacy of these vaccines. I'll just show you two very large studies in the New England Journal of Medicine. On the next slide, you can see almost 400,000 patients that if you look at the incidence of COVID infection, it's by far the lowest in mRNA 1273, very, very tiny percentage. And if you go to next slide, you're going to be looking at hospitalizations. And again, it's like even after 168 days, like 0.05% for mRNA 1273. It's a little bit higher for the BioNTech one, but that did very well also. Not only can you use it for these vaccines, and they continue to be updated with boosters, but recently Moderna announced, next slide, that you could do what's called a personalized cancer vaccine. That we call that PCV. And the idea, this is with late stage melanoma, 157 patients, you could take what are called neoepitopes, and you could take the top 34, and you could design mRNAs that will make proteins and antibodies in immune cells that will attack them. And what happened in this case is, in this trial, half of them got Keytruda, which is an immunotherapy drug, half got Keytruda and the personalized cancer vaccines. And the results were pretty amazing, 44% reduction of recurrence or death. Now, the last thing I was going to talk about is I was visited way before I did this by a very successful software entrepreneur. You see me on the left and him on the right. If you're not familiar with who that is, it's Bill Gates. And Bill has always been very interested in vaccines. And we had worked on vaccines with the idea of nano and microparticles now since the 70s. But one of the things that he and his team wondered about is, and he was concerned about people in Africa, that they didn't come back for injections. So if we could go to the next slide, I'd want to show you what he asked. Next slide. Could we come up with a single injection, but get pulses, like at time 0, 1 month, 2 months? So we'd like make a cocktail. If you look in the middle of the bottom, where you see that red box covered with gray, it says micromolded particles. Our idea is that by changing the composition of the gray part by different degradable polymers, and they'll degrade into water and CO2, it's polylactic glycolic acid, we could make them burst at different times. So these would be like little tiny eggshells, but inside the shells, they would have the vaccine, and the shells would break. And when they break at whatever predetermined time we want, you'll get those bursts. So that's what we wanted to do. And we even developed a whole new printing process. And I'm going to show you another video of how we did that. Next slide.
The stamped assembly of polymer layers, or seal method, combines computer chip manufacturing technology with a novel layer-by-layer assembling method to create an array of core shell polymer particles. First, the polymer of choice, PLGA in this example, is melt-pressed using a prefabricated silicone mold. The mold with the particles is then transferred to another substrate, where it is peeled off, leaving behind an array of polymer-based particles. These are then filled with any drug or vaccine using an inkjet piezoelectric nozzle and then dried. To seal the filled particles, a cap mold is aligned, sealed with the base, and delaminated. The resulting array of core shell particles are then removed from the base and stored until use.
So we made these, we actually published this in Science, and we published 5 follow-ups in Science, other science journals on using these for cancer and improving them and so forth. We also filed a patent on this, and let me just show you some of the data on the next slide, which is very exciting. This is going to be just one injection, if we could go to the next slide. And what you see is 11 bursts, all at different times, from like 1 week to 6 months. And you can do that by just changing the composition of polylactic glycolic acid and its molecular weights and stuff like that, because that will make the shells break at different times. And on the very final slide, what we're doing is not only testing this by injection, but also, if we could go to the last slide, we're working with Mark Prousden. It's another one of my students who's a professor at Georgia Tech. And what we're doing is we're putting them into microneedle patches. You see a close-up of those microneedle patches on the right. But the beauty of these patches is the needles don't go in very deep, but they go in deep enough so that they can do the injection, but not so deep that they hurt. So they actually look like a little Band-Aid if you go to the left and on the screen. And then in the middle, you see somebody just putting it on the skin. And our goal, is to use these for vaccines someday all over the world. That's what the Gates Foundation wants. That's what we want. And so that, I hope, will end up helping even billions more people someday. And there's all kinds of vaccines, not just mRNA vaccines, but we've been working on polio vaccine with the Gates Foundation and many others as well. Thank you so much for having me. I am happy to answer any questions.
We now invite Prof Langer to moderate the Q&A session.
Okay. All right, can you hear me? Okay, well, thank you, Bob. This is Ali Misere from NTU. Thank you for this great talk. We never met directly, but we just got a paper together last week. So I can be part of your network now. I would like to start with the first question, if you don't mind.
Of course.
You have mentioned in your talk and your paper that people would not believe in your approach and proposal. So I would like to return the question to you. Have you ever come across some technology that you're not convinced at first, and then it proved you wrong eventually?
Yeah, well, I'm sure I have, but the way I look at it, because of that experience, I have so many people coming to me with ideas, mostly my own students, but and my feeling is to never tell people you can't do something. I try to say, well, it might take a while, or or maybe you should think about doing this or that. But I don't think I've ever really, because it was so damaging to me, so discouraging to me to have all these people tell me it was wrong and turn our grants down. It really had a big effect on I think how I think about things today. That really, I don't think anything is impossible. You just don't know how long it may take. So but I'm sure there are things I probably felt might take a lot longer than they did.
Okay, well, thank you for this positive message. I'm sure many PI will take it in a very good way. Maybe I will go with some of the online questions here. So we have one here. Which one do you see here? Well, I'm not sure, but here we go. I think this is this one here. So do you think your chemical engineering background and knowledge contributed to your achievement in nanoparticle-based drug delivery?
Oh, absolutely. I think chemical engineering really was a great background for that. I mean, I learned about materials, I learned about polymers and lipids. And I also think chemical engineering, again, maybe I'm prejudiced, but I think it teaches you how to think in a very constructive way about problem solving. So I think both the specifics and the general things that I learned were very helpful. And I also think that it's funny, when I first started doing, my PhD thesis, I remember there was a really good postdoc there, Colin Gardner, and he just really drilled into me about doing things really reproducibly. He had me do standard curves with like 30 data points over and over again, so I'd get almost like 1% error. And so I think that the whole experience of doing a chemical engineering doctorate was very, very helpful to me. I have to admit, going to a surgery lab after that was like very, very different. But I do think the question is a good one, that it probably helped me a lot.
I think we have a question from the audience up there.
Morning. Thank you, Prof. Roberts. My name is Lorato from the University of Cape Town in South Africa. I'd firstly really like to thank you so much for your talk this morning. I feel extremely encouraged and inspired by your journey. I'm particularly really curious about what kept you going with the extreme level of rejection that you encountered from such a young, from your young days in your journey. What really went through your mind every time you were told, especially when you were trying to prove the concept of nanoparticles and angiogenesis, anti-angiogenic agents? What kept you going and believing in the idea that you had? And what kept you, trying out the experiments, irrespective, despite all the, grant application rejections?
Yeah, well, that's a very good question. I think there are a couple things that were probably helpful to me. One is, I think I was very lucky that my postdoctoral mentor, Judah Folkman, he kind of went through a lot of the same things. People didn't believe in what he was doing, and and yet he kept trying and he kept believing. And I think having role models is a really good thing, and he was a great role model for me. I think separate from that, maybe it's like my upbringing, my parents, I think, I grew up my dad, ran a tiny liquor store in Albany, and he worked all the time. And I think that was, a good also role model for me to see. And then maybe some of it's just genetics. I'm just a really stubborn person, I think, so but I think it's maybe the combination of all those things. But I think, like I say, for so many people, including me, having role models is very, very helpful and important.
Thank you. And just the last question, regards to the mRNA vaccine, great work, made a tremendous impact on how the pandemic eventually got regulated. What are your thoughts around access and how everything played out regarding the COVID vaccine access, particularly in African countries? Because as we know right now, I think only 30% of Africans are actually vaccinated, and there's been a lot of politics around access and patencies. What are your thoughts around that? Because for me, I found it extremely discouraging, because we have these meetings where we see great inventions and scientific innovations, but at the end of the day, when it's implementation, only certain regions in the world benefit from this great, exciting scientific innovations. What are your thoughts around that?
Yeah, well, I think that's a great question. Let me make a couple points on that. And again, you may know better than I on some of these. But, like I say, from our MIT lab, one of our big efforts, even way before this, has been exactly what you're saying. In other words, to try to, make vaccines, accessible to everybody and to ship them around the world. But you're right, when this crisis came, then it was difficult. And I don't know all of the different things. In other words, one of the things, like, for example, Moderna, it depends at what point in time. Moderna actually tried to give, I think, over 100 million doses away, and they got turned back. And my understanding, and again, like I say, you may know better than me, and other people may too, but a lot of it wasn't just the access, wasn't just getting the vaccines, it was also the journey, whether it's from the air, people would say the journey to people's arms. In other words, some of it had to do with the logistics, I guess. Even if you could get them to Africa, apparently, like I say, when Moderna tried to give them to some of the African nations, they could get to an airport, but then somehow they wouldn't get to the patient's arms. And whether it's the roads or other logistics, I don't really know. But my sense is that there needs to be more work done across the board, not only the politics and ways that you could ship them, but, and again, like I say, you'd know better than me whether there's things to do with the roads or customs or other things. Like I say, you may know more than I do.
Thank you.
Thank you.
I think what Robert is alluding to is also the issue of cold chain management, which, as far as I know, takes 80% of the cost of a vaccine. And that's sometimes the issue of going to the right location, remaining in the cold chain. I think we have some question up here.
Hello, sir. I'm from the Hebrew University of Jerusalem. Thank you for your talk. It was an absolute privilege to hear from you. And my question is a bit more technical also about the mRNA vaccine. So you're talking a lot about the technique of how the cell will be the factory and how the mRNA will be delivered, but how can we control the 3D fold and the epitope that is being presented to the immune system and prevent phenomena such as antibody-dependent enhancement of infection?
Yeah, well, that's certainly a very good question. I mean, I don't know that I'm necessarily the best person to answer that. You know, it seems to me that the vaccines have worked quite well, but how you control all these different things, I mean, it's so much of it has to do with the body's own machinery, which is what we're relying on. And of course, not everyone is the same. But I don't really have a great answer for looking at the protein folding after you get the mRNA into the cell. I wish I knew more about that.
Thank you.
We have another question from the audience.
Yes, hello. That was very fascinating. Thank you very much. Very inspiring talk. I also have a rather technical question. I mean, we just talked about, let's say, the delivery issue of the vaccines, for example, and I was very intrigued by the very last part where you show these kind of single shot injections with slow and fast releasing particles. So I was wondering, I mean, I'm really not an expert. What happens, let's say, to the slowly releasing ones inside the body? I guess they are also somewhat peg coated to prevent clearing, but where did they spend these, let's say, 1 or 2 months? And is there some kind of other clearing mechanisms that could make it complicated to be applied?
I hope so. I think the answer is I hope it won't be that complicated. We're really relying on things that we and others have done before. So the idea, just a couple of things, you know, when I showed the earlier some of that picture with all the different drugs, a lot of them were made out of what's called polylactic glycolic acid, and they would deliver in that case like anti-schizophrenic drugs, anti-cancer drugs. And what you do when you do those is you inject them underneath the skin, like intramuscular, you know, with a needle, underneath the skin, and subcutaneously or intramuscularly. And in those cases, the system just stays there, but and drug diffuses out, and the polymer in that case, polylactic glycolic acid, dissolves. But it just stays there. It just stays there, it dissolves, the drug keeps coming out. And in essence, that's kind of what we're doing here. Basically, we'd have different polylactic glycolic acids. They'd all reside in that subcutaneous or intramuscular place, but at different time points, they would dissolve completely. You know, like I say, we could make it dissolve in a week, but we could also make it dissolve in 6 months. So we're giving this cocktail, but they'd all reside at in that place in the body where you'd inject them, like you get that injection right now, probably in your arm, subcutaneously or intramuscularly. And so they would just reside there. By the way, also, just so you know, the beauty of that polymer, lactic glycolic acid, it ultimately will degrade to water and carbon dioxide. So everything goes away.
I think we have time for one quick question because there is 40 seconds left. I have one online question that I can quickly ask you, Bob. Someone is asking, do you think mRNA vaccine can be used to treat neurodegenerative diseases? What are your views on this?
Yeah, so I'll let me give you 2 parts to that answer. And I've had that question before. I mean, the question is absolutely, but I remember I was giving a talk to Glaxo's board, and somebody asked me that very question: how can we treat COVID, but we can't treat Alzheimer's? And the answer to me is we don't know the biology well enough, right? We know that the reason we can treat COVID in a way and have a vaccine is that we understand the biology. You go after the spike protein. Unfortunately, in the case of Alzheimer's, the biology is not nearly as well understood. There are other problems too, like delivery across the blood-brain barrier, but not understanding the biology you know, when you don't understand it, I don't know that mRNA or anything's going to be that helpful. You need to understand the biology first, in my opinion. So says somebody who's not a biologist.
Okay, well, on this note of, you know, supporting basic science, thank you again, Bob, for your wonderful talk and answering all those questions, and thank you everyone for your attention. Thank you.
Thank you all.